Okay, now let's take a look at the response of adipose tissue to the fasting state, which means the response of adipose to glucagon stimulation. So the job of adipose is to store excess energy, and in times of need, such as in the fasting state, to release those fatty acids so that other tissues can use the fatty acid for oxidation to make ATP. So therefore, adipose is going to have a glucagon receptor, which is, again, the 1, 2, 3, 4, 5, 6, 7 transmembrane receptor. Stimulation by glucagon, of course, will stimulate through G-alpha S, adenylate cyclase, increasing the concentration of cyclic AMP, and ultimately activating protein kinase A. Now this activation is due to the sense of we're fasting state because of glucagon, and this is telling the body we're in a negative energy balance and we need to start supplying fatty acids for ATP generation. So we're going to want to slowly ramp up the release of fatty acids. We don't need to release them all at once. So under stimulation of glucagon, just simply saying we're in a fasting state, what we will do first is to activate an enzyme called ATGL, which stands for adipose triglyceride lipase, ATGL. This particular enzyme will be phosphorylated by protein kinase A. Phosphorylation of this enzyme activates it. The function of this enzyme is to take a triglyceride, which is the storage unit of fat in the adipocyte. Recall this is our glycerol backbone with 3 fatty acids attached to it, our basic triglyceride unit. When ATGL becomes activated by phosphorylation, it will now cleave one of the fatty acids off of the glycerol backbone, generating a diglyceride. So we were going from a triglyceride to diglyceride structure with the release of a fatty acid. Now, this fatty acid is able to be transported to the plasma membrane. And again, remember, these are fatty acid binding proteins that need to transport the fatty acid in order to protect the cell from the hydrophobicity of the fatty acid. In the adipocyte, this fatty acid binding protein is called AP2. The fatty acid is then translocated, transported out of the adipocyte using the FATP, fatty acid transport protein. This is a reversal of fatty acid uptake by the cell, and this is based on a concentration gradient of high fatty acids on the inside from release to low fatty acids in circulation. Now notice that there's no lipoprotein construction here, because the job here is to get the fatty acids as quickly as we can to the extrahepatic tissue. Hepatic tissue also will be using this fatty acids, so let's make a fatty acid albumin complex. So fatty acid albumin complex is then the carrier of fatty acids in the blood and distribute the fatty acids to tissues that need it. Now, that's glucagon signaling. What happens when we get into our fight or flight reaction? Now, we saw in skeletal muscle that the flight or fight hormone, epinephrine, will signal for increased release of glucose from glycogen stores to prepare the skeletal muscles for increased activity because of the fight or flight reaction. Adipose cells will have the same issue here, and we have our beta-adrenergic receptor, 5, 6, 7. This is the beta-adrenergic. Epinephrine binds to. This will also work by activation of G-alpha S. Cascade down to adenylate cyclase, increase cyclic AMP levels even further, increasing protein A, kinase A activity even over the glucagon signaling. So notice both of these signals are feeding into one biochemical pathway. This is going to amplify the cytosolic signal, leading to increased activation of ATGL to transform our triglyceride to diglyceride, releasing a fatty acid. The next enzyme that will become activated is called HSL. This stands for hormone sensitive lipase. Hormone sensitive lipase is also activated by phosphorylation by protein kinase A. This phosphorylation event serves to activate HSL, and the function of HSL is to take our diglyceride structure and convert this into a monoglyceride. The monoglyceride will only have one fatty acid left on it, on the glycerol backbone, and this releases a second fatty acid. Think of this very similar to the release of glycogen, the breakdown of glycogen, that we are turning up the volume. So normal fasting state, we want to begin releasing some fatty acids to support tissues as glucose stores run down. But then when we really need extra energy, we want to really turn up and amplify the signal to increase the release of fatty acids, and we do this by a second activation sequence with hormone-sensitive lipase. Now we have one more fatty acid left here, and that is going to be processed by MGL, which equals monoglyceride Mg lipase. Now, current research suggests that there's no regulation at this step. So there's no phosphorylation required for activation. MGL, monoglyceride lipase, essentially becomes active when substrate becomes active, becomes available. In this case, once HSL produces the monoglyceride now becomes a substrate available for MGL, monoglyceride lipase. The job, of course, of this enzyme is to produce the final free liberated fatty acid, which, like the original fatty acids, can exit the FAT-P transporter and couple with albumin for transport, and in addition generate a glycerol molecule. Now the fatty acids are transported out in the blood. The fate of glycerol is also transport into the blood. From the blood, the glycerol is taken up by the liver, by hepatocytes. The hepatocytes now will take the glycerol and transform this into glycerol 3-phosphate. by the enzyme glycerol kinase. So we are simply adding a phosphate in the number 3 carbon of glycerol to create glycerol 3-phosphate. This can then be processed by a redox reaction to generate NADH, and in turn generate dihydroxyacetone phosphate, DHAP, in the hepatocyte. Now this is important because once we couple this with glyceraldehyde, 3-phosphate, we can generate fructose 1,6-bisphosphate and continue onto the process of gluconeogenesis in the liver. Because recall, we are in a fasting state with glucagon signaling. This fasting state in glucagon signaling also means that the liver is in a fasting state. So the glucagon will signal for gluconeogenesis, and we will take the glycerol released from the adipocyte as essentially a waste product, the backbone of triglycerides. The liver will take this and prepare it by glycerol kinase to generate an intermediate of glycolysis, but in this case it's an intermediate of gluconeogenesis because we are in a fasting state. Now the glycerol kinase is not expressed in adipocytes, and the reason for that is because if we were to generate glycerol 3-phosphate in the adipocyte, recall that this will be a substrate for the phosphatidic acid pathway and regenerating triglyceride. So during the fasting state, we want to release the fatty acids into the blood, and we prevent the resynthesis of triglycerides once these fatty acids are released by preventing the formation of the backbone. Recall the glycerol 3-phosphate that is necessary for triglyceride synthesis comes from glycolysis in a fed state in adipose. So the absence of glycerol kinase in adipocytes prevents this futile loop.